Hello everyone. Today we will try to understand dihybrid cross through a smart shortcut method. I am Varsha and let's get started. First of all, what is dihybrid cross? Many students find it so difficult to understand. It is nothing but just two monohybrid cross together. How? So dihybrid cross contains two contrasting traits that we take together. For example, seed shape and color of the seed. It's two different traits, but we are taking it together. This follows the law of independent assortment, which means the gamete formation of these two different traits occur separately and they assort separately. They are not dependent of each other. I need not to say that capital letters represent dominant traits and small letter represent recessive traits. The seed shape was capital R, which is round, small r, which is wrinkled, and for seed color, capital Y, which is yellow, and small y, which is green. Now, for the parents. Parents are always taken as true breeders. It means there is no contamination of multiple alleles into them. As you can see, capital RRYY, only one type of allele for both the character, and similarly, small RRYY, one type of allele for both the characters. So the gametes formed from them will be only two kinds, which is capital R and capital Y, and from the other parent will be small r, small y. So when these two are crossed, we do the crossing just as monohybrid cross, we will get F1 generation, which will be all identical and which all be capital R, capital Y, small y. This follows the law of dominance, which means that even in the presence of recessive alleles, the dominant allele shows its traits. Now, after F1, there will be F1 selfing to form F2 generation. What is F1 selfing? F1 selfing is crossing F1 genotype with F1 genotype. So here, capital R, capital Y, small y is crossed with capital R, capital Y, small y. And all the F1 generation has the phenotypic ratio of all round and yellow seeds. So we will form gametes by foil or fork method. If we try to understand how many gametes formed from these kinds of heterozygous gametes, it is 4 because there is number of two heterozygous gametes. So there will be 4 numbers of gametes. So capital R, capital Y, capital R, small y R, capital Y, and small r, small y. Total combination 4 into 4 is equals to 16. And this 16 is also the number of offspring present in F2 generation. This is the Punnett Square. You take one parent's gamete on the left hand side and one parent's gamete on the top side, and you cross it and see what all combinations you can make. Phenotypic ratio, we will understand the shortcut method. You can understand from this also, but it is not feasible to make Punnett Square for F2 generation in every question, single question in examination. So we learn shortcut method, which is we will treat the dihybrid cross as earlier said as two monohybrid crosses. Okay, so for R, I mean round and wrinkled, there will be 3:1 ratio, and yellow and green will be 3:1 ratio, and then we will multiply the 3:1 to 3:1, and the answer will be 9:3:3:1. So that is our phenotypic ratio for F2 generation. Now the genotypic ratio. Again, you can see it from the Punnett Square, but to save time, there is a way to calculate the genotypic ratio, which is again using the monohybrid's genotypic ratio and multiplying it into 2. So 1:2:1 gets multiplied by 1:2:1. You need to expand it and you will get 9 different genotypes. So 9 different genotypes for F2 generation, 4 different phenotypes for F2 generation, and number of offspring in F2 generation will be 16. Now for shortcut method that we were saying there, you can either multiply them like 1:2:1 into 1:2:1, or you can learn it like this. Write 1:2:1 in the starting, and then 1:2:1, and then double it for the middle three numbers, which will be 2:4:2. Again, let's see, it's 1:2:1, is to 2:4, is to 2:1, is to 2:1. The number of gametes formula, this is also very important, will save time. You do not need to use your foil or fork method to try to understand the number of gametes present in every cross. So all you have to do is take number of heterozygous pair, heterozygous pair having two different alleles for one single character. We are studying dihybrid cross, that's why I'm telling you just two different alleles for one character. That'll be 2, and you have to multiply it by 2 is equals to 4. Or you can use 2 upon n as formula, where n is the number of heterozygous pairs. Heterozygous means having different alleles for single traits. For example, the F1 generation, which has dominant as well as recessive gene for both the characters, but typically dominant traits prevail. Points to remember, independent assortment applies only if the genes are unlinked. For linked genes, independent assortment doesn't follow. Phenotypic ratio of dihybrid cross, second filial generation is 9:3:3:1, and genotypic ratio is 1:2:1, is to 2:4:2, is to 1:2:1. And for the number of gamete calculation, it can either be trihybrid cross or how many hybrid cross. Just use 2 upon n formula, where n is the number of heterozygous pairs. You just need to calculate quickly how many different alleles are for each character, write it down, multiply it by 2, and voila, that's your gamete calculation. So thank you so much. I hope you understood something from today's class, and I'll see you next time. Thank you.